Hello? There's Susan? Hello?
Hi,
Hi.
can you hear me?
Yes.
Oh, okay. Everything's working.
All right.
Amazing.
Yeah, okay, great. Yeah. It's like you're in a new location.
Yeah, I'm at my son's place and I'm babysitting his dogs
Yeah.
because it's the long weekend and they wanted to go to the lake and they can't bring all the dogs because they have three of them.
Yeah.
Yeah, it's kind of silly, isn't it? Yeah, I've got one right here.
Yeah. Yeah. Oh, great. So we have any updates, things?
Um, I don't know. I have two simulations. They're both kind of working. One I put equal force on it and it tilts.
Oh.
So I don't know what that's about.
Yeah, I don't know either. So it's the adventures of
Right.
tensegrity.
Yeah. Yes. Yeah. So it's probably just off center, just slightly or something.
Yeah. So Jiahang said he's not able to talk today. I might hear your talks. That's good. Sushmanth is there. Hello? And I know he wanted to discuss something in the Slack, or he was discussing something in the Slack in the DevoLearn Devo Graph channel. It looks like I think he got a response from Mayuk, and so I think that's something we can talk about. If we can talk about later, it's fine. But it looks like it's being resolved. So it was just about some of the issues with image segmentation and things like that. So I'm not sure if Sushmanth is there available to talk. Okay, well, we can do this later in the meeting. So the first thing I wanted to talk about was we have a couple of, I guess we'll get right into papers and things that we have been waiting on. I don't have any features today, but let me share my screen. And so a number of interesting things here. The first is this collection of papers we've been getting on curvatures and planes and embryos. And then there's this paper on uncertainties of the fossil record for diatoms. So I know Dick is interested in diatoms and sort of early life. So that was something that, well, I mean, you can watch it later. So why don't we get into the diatoms one first? I think that's really interesting. So everyone see my screen? Good? Okay. So the first paper here is uncertainties surrounding the oldest fossil record of diatoms. This is the kind of organisms we work with, the ones you've seen before, of all different shapes. The cells are all different shapes. They can be single cell organisms. They can be multicellular organisms. And you've seen the colonies of single cell organisms that move like an accordion. Those are all diatoms. And so there's a lot of diversity there. And the question is what are their evolutionary origins? And so this is a nice paper on sort of getting at the origins of diatoms in the tree of life and where we find the earliest fossil record of them. Now, they're made of this silicate. The cell walls are made of silicate. So in theory, they should be easy to preserve. So that's good that we have those around. But we don't, it's still unclear as to where they fit into the fossil record. So molecular clocks estimate that diatom microalgae, one of the world's foremost primary producers, meaning that they produce energy through photosynthesis for secondary producers, which are organisms that eat them, and they're able to produce energy. So a lot of plant species, a lot of algae are primary producers. That's how they refer to them ecologically. And then they originated near the Triassic-Jurassic boundary, which was about 200 million years ago. So this was in the time of the dinosaurs, and close to the sort of the first large dinosaurs, I guess. Yeah, which is also close in age to the earliest generally accepted diatom fossils from the genus Pyxidicula. I guess that's how you say it. During an extensive search for Jurassic diatoms from 25 sites worldwide, 3 sites yielded microfossils initially recognized as diatoms. If you remember the things we did on the embryo a few weeks back, you'll remember that it was very hard to determine what was an embryo and what was just some cells that were preserved. That's one of the problems you'll have again and again in the fossil record with things like trees and other types of parts of organisms. You have to get it into the context of the whole organism or in the context of some sort of ecological thing. So, it's exceedingly rare, for example, to find a full skeleton of, say, like an ancestral hominid or a dinosaur, but they can be found. But in this case, you'll find a diatom, and you don't necessarily know, you can't identify the species very easily. So these are things that you just have to work around. This led us to systematically re-examine published evidence in support of lower and middle Jurassic Pyxidicula fossils. Although these fossils resemble some extant radiocentric diatoms and have character states, which means that they have characteristics that may have been similar to those of ancestral diatoms, we describe numerous sources of uncertainty regarding the reliability of these records. We conclude that the lower Jurassic fossils were most likely calcareous nanofossils, whereas the middle Jurassic species have been reassigned to the lower Cretaceous and is likely a testate amoeba diatom. So this widens the gap between the time of origin of the oldest abundant fossil diatom record to 75 million years. So basically, those earlier finds, they couldn't be sure that they were diatoms. They think there maybe have been some sort of amoeba. And so now they still are sort of left with a mystery as to the earliest diatom versus the first abundant fossil diatom record, which is dated to 75 million years. So this just talks about how it's difficult to find and to discover and validate microfossils. So microfossils are these, if you recall, your average diatom is maybe on the order of 100 microns, which is a very small amount, a very small size, and usually have to identify it very carefully under a microscope. You have to sort of search pieces of rock to find these. So it's not, easy to do, and you can confuse it for other types of organisms at the time. So it's quite a game. So they talk about diatoms, and they're they were found all over the world, and they continue to be found all over the world. And they know kind of sort of the first abundant fossil record. They've been searching different candidate sites for even earlier finds, but they're not quite sure that they found the earliest diatoms, but the search continues, I guess. So this is what a diatom. This is a electron microscopy image of an assemblage. What it looks like. This is a species of diatom here. This is from a piece of rock that they've gotten, and they're doing the microscopy on it, and they show these little, they almost look like chambers or holes, and that's sort of the remains of the diatoms. So, and then this is a close-up of that where they're able elemental mapping of oxygen and carbon on a pleurozonaria specimen. So this is where they're looking at the cell wall, and they're looking at sort of fixation of different oxygen and carbon molecules going through the cell wall. So this is really small. This is 5 microns, this line. So that's several of these different pores. And so you can see how small these are. And so this is a very, very micro fossil. This preservation of this is actually quite good for the size of a feature. So this is the kind of feature that you find in diatoms if you're looking close up on the cell wall. And this is certainly, it's not something that, there's some ways you can tell different species from one another. So it's not that they don't have the resolution, it's just that sometimes there aren't the kind of characters to differentiate them. Nevertheless, this is more electron microscopy of diatoms recovered from cherts or rocks from this other site. So these are examples here. This is 10 microns. This is a little bit wider shot of this specimen. And you can see again the different samples here. And so I think they're all at this is the 10 micron scale for all of these images. So you can see that there's, they're able to identify certain aspects of the cell wall and some of the pores. Let's see. This one here is a compilation of specimens used to validate lower Jurassic fossils. So now they have to validate those fossils. They have to look at other candidate fossils. They have to know what it is. So they have to compare it with other things to make sure it's similar or different. And so they've done this with a lot of these different fossils and drawings of fossils that you'll see here. So you have all these different things that they're using to validate it. Sounds like we've got, oh, there's Dick. Okay. And there's Morgan. Hello. We're talking about fossil diatoms, actually.
Oh, okay.
So that's what those images are. Well, these aren't fossil diatoms. These are the fossil diatoms here, and especially here. These are sort of the silicate walls of diatoms, really close up.
Yeah, those are obvious.
Yeah. And so, so this is I think the timeline of their collected samples. So this is a depth in centimeters. So they're going down in depth in this at the site. They have these different samples that come from these different places. This is the number of years ago that these samples were gotten from. So from 181 million years to 182.7 million years, going down into the assemblage. And so these are the positions of the collected samples going down. This is the Posidonia shale. So this is actually their position in that shale. So this is a compilation of specimens used to identify lower Cretaceous fossils. So again, they're doing this comparative work. They have these very things from the very early 19th century on to like 1902. So they didn't have electron microscopes back then, so they use drawings of things. Sometimes the drawings are very precise, and so you can pick up features from those drawings, but they're not electron microscopy. So we have to sort of use these as guides, but they're imprecise because they're hand drawings. So this is an example of some of the specimens used to identify these. They have fossils that are available, so we can look at those as well.
Okay, Bradley?
Yeah.
In those days, they used a thing called a camera obscura. You familiar with that?
Yeah, I've heard of it.
Okay. The advantage of it is you can keep focusing through the specimen and only draw the sharply visible parts. So those might actually be quite accurate.
Yeah. Okay. Yeah, this kind of talks about this section.
Maybe you can explain what a camera obscura is for the youngsters here.
Oh, okay, yeah. So this part actually, I think, talks a little bit about some of these challenges that we have with identifying early fossils. Um, and, there are a lot of challenges with different types of assemblages of fossils and, identifying. I mean, they know how to date things, but they can't, there's this comparison between the thing they think it is and then other types of things that were around at the time. And that's the biggest challenge, I think, because you have things that look very similar. And so, it's going to be hard to really definitively say this was part of the species or not. It really does depend on these characters, on the phenotype of the organism. So in the image, you have to identify features that are unique to that species. And sometimes that's hard to do because you have the preservation isn't perfect. You have things that look very similar. And, there are a lot of things that it's hard to do. So that's one of the big challenges here. And so they put a whole section in here because I think they're not quite sure what they have. This is, despite this being, this is in scientific reports. I was going to say it's a Nature paper, but it's not necessarily the Nature. So they kind of go through some of one of the other challenges too is that you can have these reassessments of the fossil record. In other words, you can, people go back and correct things that have been found previously. So there are early reports that were made that have to be updated, and that updates the taxonomy of these different species groupings. And, even sometimes things are misidentified because for various reasons. People have different criterion for classifying things into species. And so there are whole debates about that. So as described above, the third purported diatom fossil described by Rothpletz P. annulata has all the features of a testate amoeba. Reclassification of Rothpletz's lower Jurassic fossils as the Calcareous nanofossil and lower Cretaceous fossils as testate amoeba creates broad confusion with regards to the name Pixadicula. I'll get that right at some point. Whether it is a diatom, Calcareous nanofossil, or testate amoeba. So there are all these three possibilities for what they might be, and they don't really know yet, or they still don't know. And so Pixadicula remains one of the most confusing names in the diatom literature. To fully dissect and clarify the convoluted taxonomic and nomenclature history, it appears that 1, inconsistent usage and overall poor documentation, 2, lack of literature citations to earlier works, and 3, a series of nomenclatural changes involving key species, and 4, changes in our understanding of exactly which phylogenetic lineage this species is thought to represent, and that compounds our problems. So that's what that's what I'll talk about for that paper. Let's see, Dick posted in the chat some things about.
I thought you'd be amused by Fred Hoyle's musings on this.
Oh yeah.
Fred Hoyle's right. Diatoms have been forever, not just 200 million years ago.
Yeah. Like, how far back does he suggest?
Oh, forever.
Oh.
Remember, he had the steady state universe.
Oh, okay.
Okay. This is one of these things where you take small bits of evidence, like a poor spectrum, and you say, "Oh, diatoms look like that."
Yeah. Yeah.
Anyway, I think he did 4 papers, at least on this.
Oh wow. Yeah. So, do we have any other comments about this or questions? Okay. Looks like Sushmanth is wanting to say something.
Yeah. Hi, Bradley.
Hi.
How are you?
All right.
I was like I was having a little network issues before
Okay.
back then. Yeah. You told like we today speaking about the problem I am having.
Yeah.
Actually, I have discussed with Mayuk whole thing yesterday at Indian IST 12 p.m. We two had a meeting and we discussed the whole problem, what I was facing. I will give you a small review also.
Okay.
Right now, before starting segmentation of cells or membrane, I was able to, actually segment anything model from Facebook hasn't released any code or library to fine tune the model. So it's a bit kind of hard. So I somehow managed to fine tune the model on other data set for tumor detection. I just implemented it. I have seen whether I could implement it or not. We need three things actually for segmentation for using SAM model. Those all are. Let me take out the pen.
What were the three things?
Wait a minute. I'm sharing it.
Okay.
I hope you are seeing my screen, right?
Not yet.
Now, can you see it?
Yes. Yes.
Okay, now I will start from the explanation.
Okay.
So this is this is a code I have wrote for the fine tuning SAM. I have used other resources also. I used Hugging Face transformers and their data sets to test out. Actually, we need three things to understand to give input for the SAM actually. Those three things are a ground truth mask is needed, a fluorescence image like this type of real image is needed, and a masked image for ground truth of this is needed, and a bounding box kind of thing around the segmentation where we want, that is also needed. Let me show you the just a minute. Okay, see here you can say it. I think we need three things to give a prompt to an image: a valid image, segmentation marks, or a bounding box, or this one is needed. In their code also they clearly mentioned actually about these three things. So my idea of approach right now, I have this dataset, cell tracking challenge dataset to implement segmentation. Here we have the fluorescence image, which is the microscopy image, and the ground truth of segmentation is also there. With that, we have some. Can you see here these dots?
Yes.
Yeah, these dots we can use as a prompt of XY prompt to give input for segment anything model.
Okay.
We extract this position in XY plane and give it as input to the segment anything model. We can actually use it. So this is my idea actually to implement this. Mayuk also told me another method to implement. Then he told to compare these three models, one with this fine tuning, this method using bounding box method, and I have found other library to use also for segmentation. Yeah, this library. Actually, there is a request for integrating SAM anything model as encoder. They are using there are two things here: encoders and decoders. Encoder is generally we give an image and convert into numpy array. The numpy array is converted into which we can understand. Here they are using VIT, vision transformer, as an encoder, but for decoder they are using as unit. This is another implementation, and this is working fine. So Mike, what he told me is to use this library, try and see by this also by giving bounding box. See, yeah, the box is there, right? Here we generated the bounding box around the, the segment, the where tumor is. Like that only. For our problem, we have the prompts, right? We can give this prompts, this method, and general method freezing the last layer of the network and then giving it as input. That's there are actually three methods to implement, and then he told to compare three models same data set, then extract the weights file and compare the model accuracy. That's a bit, I couldn't explain in a clear way. I'll write out a blog about these things and I will send it to you. And these are 3 implementations. I have so many doubts, but Mayuk already solved them all. He told like use this method, you can easily solve them all. Right now, I just need to implement what all ideas he given to me. Then this week I will work on these methods and I will try to implement them.
Okay.
Last was completely confused about the segment editing model. I thought just a ground truth is needed for this all, but later upon reading the paper I read, I came to know that at least we need to give a bounding box or some dot kind of things to model to segment properly. So my goal to extract this X5 points in 2D and give it would be fine. He couldn't attend these weekly meetings, but he was closely watching me up in the Sundays or Saturdays and discussing with me. He was solving, he was able to come, I was able to communicate with him and keep track on this GSoC work. That's all my activities. What could I say? This week I couldn't work more because understanding this paper took me a lot of time. And I was seeing other resources also. When the coding period begins, I will start coding up. But actually, there are my exams from May 28th to June 13th.
Okay.
First, I will give a trial run whether I could manage both of them or not. If I couldn't manage, I will let you in the Slack or somewhere.
Okay.
I will, for 1 week, I will work on my exams and I will come back and I will start coding again.
Okay. Yeah, I think this is, I can't remember the length of the project. I think it's a shorter project, so it's, 20 hours a week anyways, so it's not full time. So, if you have to push things to another week, you'll still be on track probably if you, it's not like you have to do, yeah.
This is 350 hours project, right? This is not short term.
Oh, okay. It isn't. Well, yeah, it's fine. I mean, we can even extend it at the end if you need to, but
Just, I don't know whether I could manage my both exams and this work.
Yeah.
I will give a trial run to it. If I could manage, I will just work on it. If I couldn't manage, I will let you.
Okay, that's good. Yeah. Great.
Yeah,
Thank you.
I'll just skip the exams.
Yeah. So
I will try.
Oh, go
I
ahead.
will try to write a blog about whole community bonding period. I will share it to you. You can just read it and understand what all I did for this week, complete week.
Okay.
I will start probably by next week. I will send you the blog, etc. to you. And this will be hosted in DevoWorm also. DevoWorm blog section is there, right? I will post them there, so everyone could read them and read that.
Sounds good. Thank you. Yeah, I guess the question and comments I had were about the prompts. So yeah, you need a bounding box or like dots, which would be like the centroids, as I understand it, and then that would be like a key, like sort of it would tell it where to sort of look for. Yeah. And so yeah, we've done a little bit of that in the past, like with masks and that, and it's it's a little I mean, I guess it wouldn't be that challenging. So you plan to make these from like other, like just segmenting the data, getting like, well, I guess you have fluorescent images in some of the data sets, so that already provides you with points, like basically centroid points.
Using images, we can generate these points and bounding box. But I was trying to write code and generate them by using it. I try out both which method is perfect. I will implement that.
Okay. Yeah, it should be enough to create a mask, like from some of the data sets, because I mean it does basic segmenting. It'll give you like a map. I guess it'll give you an image set of coordinates, and it'll give you basically the places where it thinks the centroids are, and then that would be enough for a mask. Yeah, so it sounds good.
I need to discuss with Mainak actually. The cell lineage population model was developed by him. We are updating whole DevoWorm. So I don't have any knowledge regarding lineage, cell lineage analysis or some kind of stuff.
Yeah.
So I scheduled a meet with him maybe this weekend or coming weekend or middle of the week. I'll discuss with him. I don't know whether it is a classification type of problem or what kind of it is. I was trying to read his code, understand it, but it's like bit. I need to learn some other more things. So I started reading the other papers regarding cell lineages stuff to understand it properly. And maybe by this weekend I will have a meeting with him and I will understand about cell lineage also. I'll try to upgrade that one also. In these days, I was thinking to update only cell membrane and cell nucleus perfectly. But after seeing cell lineage, I got a little more interest. I will try to implement that also. I need to just discuss with Mainak, not Manik. He gave me appointment around this weekend or maybe middle of the weekend. I'll discuss with him and I will
All right. Yeah, that sounds good. That'd be good to draw from that expertise. Yeah, I mean, the cell lineage in C. elegans, of course, we know what cells are part of each cell. They have their own identity and they're part of the cell lineage. In other organisms, oftentimes you'll have a case where the cells will sort of form lineage trees based on their starting point. In other words, you have a lot of cells that can be any cell in the lineage tree. And so, like in mouse, for example, mouse embryos, they've tried to construct the lineage tree, but you have cells that can be like a certain cell in the lineage tree, and it's not necessarily traceable to any one cell in a position. So there's this sort of interchangeability of the stem cells that become those cells. So in C. elegans, however, we don't have that problem where basically the cells will divide from like a 2-cell state. You'll have the AB and the P1 cell, then you'll get divisions that are in certain parts of the embryo, and it's always that cell. So we have like it is almost like a classification problem in that you can find a cell in a certain position in the embryo, and you kind of know that that's that cell. You can also trace from like a 2-cell state and trace out where they should be because they're always usually in the same place.
Bradley?
Yeah.
Yeah, I've got a question about C. elegans.
Yeah.
Sometimes it produces about 50 pairs of supposedly identical cells.
Right.
Are the trees from those cells, a pair of cells, identical?
You mean like the bilateral cell pairs?
Yeah, whatever. I don't know.
Yeah. I think, well, more or less they are, but there's some differences, and
They can be different.
yeah, they can be different, and especially like in post-hatch or post-embryonic development, you get like you even get like this sort of regulative development in some cells where it produces like in the seam cells, for example, in other places, but that's limited. But mostly the bilateral pairs they divide and they come out on like the right and the left side, and you get like basically very similar. They're very similar. They may do things if they're in the
interesting, but they can diverge a bit.
Yeah, well, like for example, if they're part of the connectome, they're going to have maybe different functions. But sometimes that's they have this sort of symmetrical aspect where you need to have symmetrical cells in a circuit to do forward and backward movements or something like that.
Okay, interesting.
If you have any papers regarding cell lineage, please share it to me. I was thinking to read it.
Oh yeah, I can do that.
My new images of axolotl eggs, which are kind of blurry, are about 200 megabytes. So I'm not sure where I should. Well, I'll send a little stick to you, Bradley. Dick? Should I send you a stick too? Axolotl images.
I think I'll wait for the sharp ones.
You'll what?
Wait for
the sharp ones. Oh. The problem with this batch too was that they somehow they didn't develop. I had about 6 of the eggs out of the entire batch, which is over 100 eggs, developed. The rest didn't.
That's
So,
weird.
yeah.
Usually it's the reverse.
Anyway, so they weren't too viable, but they were good for trying out the microscope, I guess.
Okay, good. Okay,
Well,
Bradley, I put an abstract in the chat. This is for a paper I've been asked to review that's over my head.
Okay.
I figured somebody with Sushmanth might be able to handle it after his exams.
Yeah. So this is the reproducibility in radiomics.
Yeah, it's a bunch of, but it looks like they're trying to do segmentation, see how good it is.
Okay. Yeah.
So it might be worthwhile. It might stretch somebody's vocabulary, but
Yeah.
Okay.
Yeah.
These are some CT scans about what they are.
yes, and you guys are experienced with segmentation stuff, so you might be able to critically assess this paper.
Yeah. Actually, I did some segmentation on CT scans. Actually.
Okay. Well, if you're interested, send me your email and affiliation, and I'll forward it to the journal.
Okay.
Okay.
I will work on this after GSoC maybe. I mean, right now this is not a. I think it's good. I mean, I could implement it. I'll send you my mail.
Okay.
Okay.
Yeah. And Susan, I'll send you my address for sending those images. If was that okay? Email. Yeah.
It's okay. I have this off because the dogs.
Oh yeah. Right. Yeah. So that's good. Thank you, Sushmanth, for the update. Yeah, it looks like it's pretty. Looks like it'll be a pretty good thing when you get everything set up, and pretty nice improvement to things. So, did we have anything else people want to talk about before we move on, or?
That's it. I mean, just a paper. I need to understand cell lineage. That's the only problem. Because when I see these cells, there is some something called PLAPPAP cell. Some different kinds of cells are there. I don't know what they are. I'm figuring out like A cell, B cell, B cell, B cell.
Oh
But
yeah.
I don't know about this PLAPPA continuation of that. AB, PLA, PP, AAA.
Yeah. So yeah, I can send you some references on that, but basically that nomenclature is, part of development where you have these two cells that diverge initially, AB and P1, and then AB will divide into APA and AP, APP or something like that, APL and APR, I think. And the point is that there's this orientation. It's basically nomenclature that gets tacked onto AB as the cell divides. So the nomenclature is, if you see an L, that's left. If it's R, that's right. So it goes to the left side or the right side. And then A and P are anterior, which is forward, and posterior, which is backwards. So the cells will divide like that, and they'll move around to those positions. So I can send you. Oh yeah. So I can send you. There's a like a nice interactive app that shows you like the positions of all those. So it runs you through the lineage tree and gives you the different cells as they're named, and then they're So that should work.
That's it. Those are my doubts, and I will give you update this blog thing by this weekend. I'll start coding also.
Right.
Coding starting maybe May 28th. This is next week. Coding starts actually officially. We need to start code. Before that, I will write a blog. I will give you what all work I did in my community bonding period, and I will attach you with the resources I've got, and I will start implementing the code.
Okay, sounds good. All right, thank you. All right, so anything else? So this is the other thing I wanted to talk about was we have a bunch of papers on curvatures and planes in embryos. And when I mean curvatures and planes, I mean this geometry of the cell. So we've been talking about like curvatures, and we've been talking about like some of the role that curvatures play in morphogenesis and some of the physics of the cell. And then the planes are like these planes that are flat surfaces. It could be different planes formed by cells or through the cell or whatever. So these papers, let's see, where do we want to start? This one is collective curvature sensing and fluidity in three-dimensional multicellular systems. So this is a Nature Physics review article. This is about collective cell migration and talking about some of the aspects of what it does, what it's useful for. In embryonic development, you get cells that move around the embryo to different places after dividing or as they're differentiating. You have wound healing, which is where cells have to come to the wound and close it up and form like a mesh where it can keep the wound from getting infected. And then tumor metastasis, which is where tumors form and grow. And so all these things involve collective cell migrations in different ways. Substrates or interfaces associated with these processes are typically curved. So that's interesting that, that there's this curved interface that is necessary for migration.
What's the title of that again, or the
collective curvature sensing and fluidity in three-dimensional multicellular systems.
Oh my goodness! you don't have it on screen, so I just.
Oh, I'm sorry. I didn't share my screen. This is like embarrassing. Okay. All right. So this is the paper. Collective curvature sensing and fluidity in three-dimensional multicellular systems. So this is a Nature Physics paper. And so this is what I was reading from here, the abstract. So it requires this sort of curved interface or substrate with the radii of curvature comparable to many cell lengths. So this is where, it's multiple cells migrating, they're on the surface that's curved, and they're all sort of on the surface at different locations. So the radius is, pretty large compared to a single cell. Using both artificial geometries and lung alveolospheres, which are these little alveoli, which are the little air sacs in the lungs, and I guess these are little maybe organoids that are based on these little parts of the lung, derived from human-induced pluripotent stem cells. So yes, they use induced pluripotent stem cells, which are these cells that we reprogram from, say, like some source cell. It becomes a stem cell, and then we can differentiate it in different ways. Here we show that cells sense multicellular scale curvature, and that it plays a role in regulating collective cell migration. As the curvature of a monolayer increases, so as we increase the curvature of this interface, cells reduce their collectivity and the multicellular flow field becomes more dynamic. So this is where, you're changing the curvature of this monolayer and it affects the behavior of the cells. Furthermore, hexagonally shaped cells tend to aggregate in solid-like clusters surrounded by non-hexagonal cells that act as a background fluid. So these are, where if you have hexagonally shaped cells, they can form clusters. You can get other cells surrounding them that can act as a background fluid to move them or to move around them. We propose that cells naturally form hexagonally organized clusters to minimize free energy. And the size of these clusters is limited by a bending energy penalty. So they do this sort of energetic analysis of migration, and they, basically assume a minimal energy for these hexagonal clusters. We observe that cluster size grows linearly as the sphere radius increases. So you have this sphere where there's this radial surface. As the sphere radius increases, these clusters of hexagonal cells grows linearly, so they just grow in size as the sphere radius increases, which further stabilizes the multicellular flow field and increases cell collectivity. As a result, increasing curvature tends to promote the fluidity of multicellular monolayers. Together, these findings highlight the potential for understanding curvature as something that regulates spatial and temporal characteristics of these systems. So they explore a number of things here. They're looking at cell migration, they're looking at cell jamming, which is where cells are flowing, and then if they get to a certain density, they stop flowing. They also are exploring this idea of packing geometry, which is, if you have a bunch of things that are hexagons, they're more efficiently packed than things that remain spherical. And so those are some of the things that they're exploring here. For example, epithelial cells growing on flat substrates approach a jamming transition during maturation, exhibiting an increasing velocity correlation length, decreasing cell speed, and decreasing cell aspect ratio. So that's as these epithelial cells flow on a two-dimensional surface. They can have this jamming phase transition where they, at a certain density, stop flowing. And you can sort of measure out those things. In contrast, most biological structures, such as alveoli, bronchi, and intestines, so these are parts of the lung and then parts of the intestine where the surface is extremely curved, are naturally curved in 3 dimensions. Even for inert materials, it is known that curvature fundamentally changes basic processes such as crystallization. Yet it remains unclear to what extent the cellular collective response is to curvature itself. So what does it mean that the cells are behaving on a curvature instead of a flat surface? So, they go through a lot of different things where, there's a lot of variability at very small scales here. So we have to kind of, we can't assume anything from the 2-dimensional case. So let's see if there are any pictures in this. So this is a curvature here. This is a concave curvature or convex curvature. And then this is the radius of that curvature. So you can see that you have this cup or this sort of dome that comes up from a substrate, maybe a 2-dimensional substrate that, this surface has 2-dimensional. This is 3-dimensional because it has this curvature. You can see that these are cells along this curvature. These are the nuclei of the cells. This is a fluorescent image. So you see the green nuclei here, and they're on this curvature. So you can see that they're sort of distributed in different ways around the curvature. It's not evenly distributed. You see these little clusters, and these are the ones that they're referring to, I think, with respect to the hexagonal versus other cells in the background. These are MDCK cells. They're grown on different curvatures, and this kind of shows their collective behaviors in the aggregate. So they're not really analyzing it in depth here. But in this case, they're doing some modeling. They're looking at this 2-dimensional surface, and they're looking at the spherical surface, and they're actually able to change the radius of this curvature, and they're able to see some divergence here, and then there's a distribution of divergences. So the multicellular flow field for these MDCK cells is more dynamic with a larger curvature. So the larger the curvature, which is this increasing radius of the substrate, leads to more divergence. And so then you have this, additional cases here where you can show the curvature decreasing and look at some of the effects of that. So this is this alveolosphere, which is derived from human-induced pluripotent stem cells. This model to confirm. So this is the model here that they're using, and they've stained it for different things in the cell. And they show this extracellular matrix and then the curvature here. And they're showing increases and decreases in curvature. They confirm that collective cell behavior is strongly affected by curvature, as they showed in the theoretical model. So they're able to show this in a biological context. And so they're able to write out some equations here where they can look at the total energy to form a circular hexagon pack. Then they look at the 2D Young's modulus of the cell pack, which factors into this calculation. By minimizing the delta G, we obtain a critical pack size, which is linearly proportional to R. So they're able to do these, kind of calculate out a sort of a scaling almost. And so this figure, they have curvature-induced bending energy causes higher dynamics in the multicellular flow field. So from the equations we just saw, now they're able to calculate an energy flow field or an energy minimization scheme. And here they show some of these cells, hexagon cells versus non-hexagon cells. And they can show with differences in size and curvature in the alveolospheres and in the simulation what those look like. So you see in the alveolospheres, you get a similar result to the simulation. And this just shows different conditions in the alveolosphere and the simulation. So this is a case where there's high shear in this part of the cell, in this cluster. And you see that there's sources and sinks. And so what they mean by sources and sinks, let's see if they talk about it in the legend. I don't think they really talk about it too much. But basically you have these different forces acting on the structure, and depending on the curvature and depending on the different clusters that are in here, they deal with them differently. So yeah, that's an interesting paper because they really kind of put a lot of that curvature, they put it to the test. They look at it in a biological system and in a simulation.
Okay. Bradley, there's something very strange about the results.
Oh,
Can
yeah.
you bring up one of the pictures again?
Oh, yeah. Which one did you want to look at?
The last one you showed.
Okay, so that's probably figure 4 or something. This one here?
Short. Yeah, that's fine.
Okay.
Okay. Now, blow it up, please. Okay. What's strange about this is the following. The highest curvature for these cells is at the boundary with the plane.
Okay.
And yet, I can't say, there's some perspective problems here, but I can't say that there's anything special about extremely high curvature.
Yeah. Yeah, it's hard to really see where the change
if you
is.
look
Yeah.
at the bottom ones there, high shear and whatnot, the highest curvature is right at that circle boundary.
Yeah.
And yet nothing special happening there.
Yeah.
Okay, so this seems to be an internal contradiction in the data.
Huh. Yeah. Yeah, I'm not sure what the because these don't look that different as you go.
Oh.
Yeah.
Well, as you go towards the circle boundary.
Yeah.
These sketches fine because you're seeing them in perspective, so the things get closer.
Right.
Okay, but it's not obvious.
Yeah, it's not.
But something's wrong here.
Well, yeah. I don't think, they also don't test it in different. It looks like it's a constant curvature. It's just in this case, they're increasing the size. So
Yeah, but it isn't constant. The highest curvature is right at the circle, no matter what size.
Oh, I guess just, across the surface.
Yeah.
Okay.
Okay.
And curvature should be the same throughout.
Yeah.
Right?
Well,
Throughout the sphere.
Well, to do that, you need a sphere, not a hemisphere.
No, well, this is a sphere.
They're showing a hemisphere.
Oh, they're showing a hemisphere.
Yes, that's why. Contradiction.
I need to use this paper as a quote. So.
Yeah, well, you may be more than a quote. I think criticism is important.
Yeah.
So
did they even ask the question, Bradley?
Well, I'm not sure. I haven't read through in detail.
Okay, I think the curvature at the edge is the highest.
Yeah.
That's obvious.
Yeah, I don't know if they're meaning to say that this is a sphere or a hemisphere or what, but
Oh, it's a hemisphere, but in your original picture of how it's made.
oh, okay.
You know, you said it's a flat surface with a bump.
Yeah. Well, I don't know though in the simulations or well,
Well,
yeah.
okay. Simulations are one thing, and reality's another.
Yeah. Well, anyways, yeah. Well, yeah, it would be interesting to see exactly what the so that yeah maybe this the other two papers might give us a little bit more insight into what you know what the role of curvature is. So this paper is curvature induces active velocity waves in rotating spherical tissues. This is a Nature Communications paper. So um, the abstract of this is the multicellular organization of diverse systems, including embryos, intestines, and tumors, relies on coordinated cell migration in curved environments. In these settings, cells establish supracellular patterns of motion, which means multiple cells engaged in collective motion, including collective rotation and invasion. So they move into new areas where they rotate in a synchronized manner. While such collective modes have been studied extensively in flat systems, geometry and topological constraints on collective migration is largely unknown. So they discover a collective mode of cell migration in this paper, rotating spherical tissues, manifesting as a propagating single wavelength velocity wave. So this is a wave that they're proposing in that spherical tissue in that collective cell movement. So this wave is accompanied by an apparently incompressible supercellular flow pattern featuring topological defects as dictated by the spherical topology. Using a minimal active particle model, so they're using this a modeling approach again, we reveal that this collective mode arises from the effect of curvature on the active flocking behavior, which just means that they're behaving collectively according to interaction rules of a cell layer confined to a spherical surface. So their results identify curvature-induced velocity waves as a mode of collective cell migration, and this impacts the organization of 3D tissues. So again, here's an example here of the nuclei in this sort of collective, and there's the spherical pattern. You have the extracellular matrix out here. The cells are migrating around this axis, so they're migrating in a sort of a spherical pattern, but it's organized. Here, these are examples of collective migration along an orientation route. So they're all kind of collectively moving around each other, but they're going in this direction or this direction or this direction. So you have this directional migration. And they kind of explore things like angular speed, rotational order, and then they look at the distribution of this over their model versus their experiments. So they have this rotational order that varies across. So it's not like a deterministic process. So this is a stochastic global rotation of a spheroid. So this is a spheroid. Here are different examples of it. These are the nuclei. These are the individual movements of the cells in the spheroid. Then this is the movement of the spheroid over as a collective. So they're able to do this in a simulation and in an experiment. So they kind of show this distributions of angular speed and rotational order, both distributions across time in different spheroids. So these are distributions. This is, I think, angular speed and rotational order seems to be a little bit different, but that's how they characterize that. So this figure here is these are velocity waves in rotating spheroids. So now you have these velocity waves that go across. This is the sphere here with the different, as I said, it's axial. So you have these poles, you have the equator, and then these are the cells within this sphere moving around. You have, I guess this figure D and E are Chymographs of the azimuthal component of velocity fluctuations around the equator of one spheroid. I guess this is higher, this is lower, the blue is lower, the red is higher. So you have this over time, you have this shift in angular velocity, I guess, or angle, and this variable. So the red shifts over in this direction. So it just shows that there's this change in this wave that goes on, this change, I guess, in rotational momentum or something throughout this spheroid. So you can see this pattern in the heat map here. As opposed to E, where you don't see this, it's just kind of constant across time. And then in this figure, you have supercellular flow patterns of the spheroid surface. So these show the flow patterns sort of broken down as the way in which they're moving. So you have these different colored arrows. You have vortex defects and saddle point deflects, and in these, in the experimental data and model. So that in this way, they show the flows, the flow fields, and then these different defects, saddle point and vortex. So let's see. So A is the snapshot of the tangential velocity fluctuation field in the surface layer of an individual spheroid. So that's A. This is where you have this flow field. B, flux analysis around the saddle point ahead of the velocity wave maximum. You have to go to the method section to get like precise definitions for a lot of these things. Bar plot shows the absolute value of the average influx, mostly along the equator, and outflux mostly towards the poles. So things come in at the equator and move out towards at the poles. So you see that up here you have, this is the bar graph they're referring to. You have this influx and outflux, and then this is what it looks like in the sphere. So you also have backside and front side of the average velocity fluctuation field. So that's C and D. That's up here. So you can see the influx and outflux. And then this is F, G, H, and I are all tangential components of the average velocity fluctuation field in spherical coordinates for the experiment in our model. So the experiment versus the model. So then this shows more of the same. This kind of shows velocity waves in a model of active particles on a sphere. So these again show these waves and evidence of the waves. This shows noise amplitude versus alignment strength. This shows that collective rotations are one part of this phase diagram. You have a region that's a region of disorder, a region of quiescence, and then the collective rotations. It's usually at this negative alignment strength, or I guess for values 10 to the negative 2, 10 to the negative 1, which is, down in this part of the measure, and then noise amplitude, which is 10 to the negative 1, or from 10 to the negative 1.5 maybe, down to 10 to the negative 3. So it's in this region of their phase space. The point is that there's this difference between collective rotations, disorder, and quiescence, which means that the cells are not moving. And so it's sort of this order at the edge of chaos idea shown in data. And yeah, so this is just showing more of the same, I think. And then the discussion, they just kind of say that they've discovered this collective mode of migration. Wave-like instabilities are a key feature of collective behavior. The wave-like phenomena of multicellular systems include mechanical and mechanochemical waves and spreading epithelial monolayers. More broadly, sound waves have been theoretically predicted and experimentally observed in active matter flocks on flat surfaces. So in active matter, which doesn't need to be biological, it can be just like particles and things like that. You get these kind of flocks on flat surfaces. On curved surfaces, you get maybe this flocking behavior with a much more dynamic set of behaviors. Hydrodynamic theories predict such sound waves to also manifest on spherical surfaces, where hydrodynamic instability causes velocity waves to propagate in phase with density waves along the equator of the rotating flock. So this is something they find more generally in spherical systems that have hydrodynamics, and so you'll see this sort of these instabilities and these waves operating there as well. And so finally, as an alternative to sound waves, the active velocity wave may be related to kinematic waves, which are waves of motion, where you have waves that are sort of have a motion component produced by the advection of a fluctuation pattern by the background flow. So this is an example here from a sphere, a truncated sphere, and a cylinder. You have this closed sphere, the truncated sphere, and the cylinder. And these are the signatures over time. And you can see that the sphere and the truncated sphere show, more sort of dynamic behavior than the cylinder, where it's just in this one orientation. And they're able to migrate around freely, but not on the spherical surface, which provides, this sort of curvature. I guess this is a case of constant curvature. So this is not a hemi sphere. So, so that's that paper. And then the final paper is...
Oh, Bradley,
yeah.
Before you go, okay, a couple of comments. I've seen these patterns in at least 3 places. If you look at anybody's head who still has full head of hair, they tend to have whorls on the top.
Yeah.
Right? And that might be related. Another one is convergence extension movements during neural plate formation in embryos. I don't know if anyone's looked at the curvature effects of that. There might be some.
Yeah.
And the third one, which they bring up, is the flocks. Now, the flocks have a very interesting feature. If you watch movies of murders of birds, okay?
Yeah.
They have a sharp edge to them. And I wonder what the relationship between the sharpness of the edge and these slope patterns might be.
Yeah. I'm not sure. Yeah. But yeah, the edge of the flock, you mean?
Yeah, the edge of the flock is usually a very sharp boundary, even though it's dynamic.
Right.
But it's sharp.
Yeah.
I haven't seen an explanation for that sharpness.
Yeah. Yeah, I don't know. I'm sure there's a whole literature on flocks in soft matter, which I'm not familiar with, but I would think they would address it there, but you don't know.
Yeah, no.
Yeah.
Okay.
All right. Well, yeah, that's good. That's good to think about. And I wanted to bring up the waves specifically because we talk about that in embryos, and I think it's interesting how anyone proposes a wave, what that means. So the last paper is redundant mechanisms and division plane positioning. So this one isn't like the other two. It kind of talks about this sort of plane positioning. And this is in plant cell division. So this is a little bit different set of systems here. So redundancy in plant cell division contributes to the maintenance of proper division plane orientation. Here we highlight 3 types of redundancy. So there's temporal redundancy, or correction of earlier defects that results in proper final positioning. There's genetic redundancy, which is the functional compensation by homologous genes. And then synthetic redundancy, or redundancy within and between pathways that contribute to proper division plane orientation. And so understanding the types of redundant mechanisms involved provides insight into current models of division plane orientation. And they don't talk about spheres in this, but they talk about the different plane orientations. So what is a plane orientation? It's where you have this process of cell division. You get cells that divide into daughter cells, and then they form this plane. So we talked about C. elegans. C. elegans develops along this anterior-posterior plane, the anterior end being like the AB lineage, the posterior end being this P1 lineage, and then there's differentiation along those different lineages, but they all proceed along a plane, which is the AP plane, and then there's a second plane that emerges, which is the left-right plane, where you get this bilateral symmetry of cells. So you get this ability for symmetrical cell divisions. You get, along the left-right plane, you get non-symmetrical cell divisions in different ways too, but largely you want to preserve that plane orientation. But even the anterior posterior plane, you have different things that emerge. So for example, in C. elegans, you always get the reproductive structures towards the back end, the connectome cells that form the main central nervous system towards the front end, and you don't have it's not randomly placed. So this is the importance of these planes, that they provide this sort of template for things, that they end up in generally the right place. So plant cells do not migrate and instead control the location of new cells by positioning the division plane. So in plants it's different. You have this plane orientation, which is uniquely unique in plants, but you see this in all organisms to some extent.
Okay, Bradley,
Yeah.
I'd like to challenge that statement.
Okay.
If you look at a plant meristem,
All right.
where there's very active growth, it's clear that the cells have to change positions. But nobody's investigated as far as I know.
Yeah.
Okay. So
there's convergent extension type things going on in meristems, as far as I recall. Yeah.
Okay. Another question is is the apparatus that does cell division in plants the same or quite different from animals? I forgot.
Oh, I've forgotten too. I think it's similar, but I don't think it's vastly different, but still.
The spindle apparatus.
Yeah, I'm not sure.
It's similar.
Yeah.
It is similar. Okay.
Yes, it's just plants tend to use their cell walls as part of their mechanics.
Yes, and that's what I'm saying. In meristems, the cell wall is still pretty flexible.
Yeah, it's more embryonic.
Yes. Okay. Sorry about challenging the first statement there.
No, that's fine. Yeah. So they kind of go through some of these aspects here. Actually, they talk a little bit about the spindle. Spindle captures and separates chromosomes during metaphase and anaphase. Spindle formation is reviewed in these places. After chromosome separation and anaphase, the spindle disassembles to form plant-specific structure. They also have things like microtubule arrays that contribute to the positioning of this specific structure. This specific structure also directs the formation of the cell plate. So there are all these different parts of cell division that contribute to the formation of this plane and the maintenance of the plane. And so, you'll see, for example, in mutants, you'll see differences, but cell elongation prior to division typically favors a division bisecting the long axis of the cell. The relationship between division, plane orientation, and cell shape is discussed in these references. Identifying mutants that only alter PPB formation or positioning, some of which are discussed below, but do not seriously alter interphase microtubule orientation, polarity cues, or cell shape, provide exceptionally valuable insight. So what this means is that there are a bunch of things in cell division, a bunch of aspects of that are sort of set up this plane. You have different microtubule arrays that get set up in response, and this sets up this sort of plane of these planes that we're talking about. In mutants, which are organisms that have defined mutations where these things are disrupted, you can have that, but generally, we see that cell elongation favors this axis, and then the division, it reinforces the axis, and then the microtubules actually are also involved in this, in forming this axis and reinforcing it. And so this is an example here. You have, this is the midplane, the interphase, the prophase, the metaphase, anaphase, the telophase. So you see that the microtubules are stretching out, and then you get this replication of genetic material. You get then the two cells that form, and then cytokinesis. And then this is the cortex, which is part of the microtubule structure where it stretches out during these divisions. So you see this orientation reinforced.
Okay, Bradley, could you go back to the picture a second?
Yeah.
Yeah, some diatoms do things quite differently. The nucleus is shaped not as a sphere, but as a torus.
Okay. Oh, yeah.
And the microtubule, there's a microtubule bundle down this perpendicular to the torus, down the middle of it.
Yeah.
Okay, so it's a quite different picture for this.
Yeah.
And I don't know if people have looked at orientation in cell division of diatoms or not.
Yeah, I don't know. Okay, so this is a map of temporal redundancy. So you see some redundancy with respect to time, and they kind of show examples of correctly oriented cell divisions and when mitotic structures deviate from the correctly oriented position. So you get these mechanisms that kind of reinforce the normalcy of it. You get these opportunities or instances of incorrect orientations, but they can be corrected by this redundancy to make sure that they fit into this correct orientation. And then this is protein recruitment, so you get these aspects of protein localization that gets reinforced by this redundancy. Later and then you get later recruitment to the same location. It has a different effect. So, yeah, so this is different from the curvature aspect, but this is how we form these planes, which, if you think about the differences between planes and curvatures, the planes are sort of reinforcing these positions of cells and sort of the overall structure of an embryo, whereas the curvature is allowing for a lot of different types of morphogenesis to occur and a lot of migration to occur. So, yeah.
Bradley, there's a mechanism missing there.
Okay.
Go back to that picture with alternative mechanisms.
Let's see. Not that one. Next one up, I think.
Oh, next one up. Oh.
This one here?
Oh, I think before that.
Okay. This one here?
Yeah.
Okay.
Okay, what's missing is in Archaea, there's a strange paper which suggests that the plane of division is determined in polygonal Archaea by a Turing mechanism.
Okay.
Okay, which doesn't appear here. And I'm not sure what the biochemical basis for the Turing mechanism is, but it's a curious paper.
Huh. Yeah. Is that something that you only see in Archaea, or did they propose it's...
I don't know how universal they made it.
Okay.
They see it in triangular Archaea.
Yeah.
If you've got a triangular cell, you can't easily divide it into two.
Yeah.
Okay? They tend to divide into two, become rectangular cells, which then switch over to being triangular.
Right. So that,
okay?
yeah.
So I'm just saying, throw a little doubt in here as to what is complete.
Yeah. So that actually, goes back to the other papers where we were talking about these hexagonal cells versus the other cells, and they had different behaviors. So yeah, I think that's it for this paper. I don't know if there's any more to say about it. It's just that, this is a nice contrast between these two things, and I think they're both important for some of the topics I've been talking about physics and the physics of differentiation. And so a few more thoughts and comments on the curvature papers. So a few more thoughts on the curvature papers. It was mentioned that, is this a constant curvature or is this a hemisphere? And so what that was referring to was, in the paper there was a figure where they talked about a hemisphere, both sort of on top and on the bottom of the, so you might have like a hemisphere sticking out of a surface here, a two-dimensional surface, or sort of the inverse situation where you have something that goes down into that surface. So that was the hemisphere. And the idea being that the hemisphere won't have a constant curvature because at some point it will sort of stop at the two-dimensional surface. I don't know. I mean, I guess it depends on whether you're modeling it as a sphere independently. Sometimes if you have something like a tumor or like an alveoli, they're kind of embedded in strata of extracellular matrix or other tissues, and so they form that sphere, but they're impacted by its surroundings. The other thing is that sometimes cell balls or things that are packed into balls don't have a constant curvature. So sometimes they look a little bit like this, where you have a lot of like rough edges to that packing. So, you'll have you can break it off into pieces like this, maybe one or two cells in each of these divisions. And that's what it looks like. So there's no constant curvature. If you go to the surface of this, it's actually quite a rough curvature. And so we can't assume a constant curvature for something like this. Nevertheless, though, sometimes there are very solid spheres, and sometimes they have a little membrane around them. It's worth considering that, what are the sort of the things that drive through the uniformity of cell packings. So if we have a bunch of hexagonal cells packed very tightly, we're more likely to have a constant curvature than not. And then when we talk about planes and curvature, this is the idea here is that if I take an embryo like this embryo would develop along two planes, at least. The first being this anterior-posterior axis. The second being this left-right axis. And sometimes there's an anatomical imperative for a dorsal-ventral axis. And so a lot of times, some of the things we've done with embryos, embryo networks, lineage networks, things like that, we often have these three dimensions. So in our some of our literature, some of our papers, we have like a five tuple or a 5-dimensional equation, which is basically XYZ, which is the XYZ coordinates, time, and this factor, which can be angle, it can be other properties, and we can even have more variables as we see fit. So you might have another set of variables here where we have different aspects of morphogenesis, different aspects of the phenotype, and so forth. But these three are related to spatial location. This is time. This is usually angle or some sort of usually division angle. So when a cell divides, it divides at a certain angle. We can characterize that. We can also characterize angle in terms of these three dimensions. So these three dimensions form a volume instead of just like three independent dimensions, and we can give more information about it here. So this is the way we kind of think about it in our group. And this relates to these three different planes that you see here, and then ultimately relates to the amount of curvature that you'll find in a three-dimensional volume. We can also, we can characterize things like roughness in fractal analysis. Oftentimes they have a parameter for roughness. It's a fractal scaling factor, and that will tell you kind of this how rough the surface is. If you've ever heard of this anecdote about measuring the length of a coastline, at one scale the coastline appears smooth, but when you get to smaller and smaller scales it appears more rough. And you'll see this with these spherical packings as well. If you're interested in sort of the organismal level, you might consider it to be a smooth spherical packing with a smooth surface. If you're considering this in terms of single cells, you'll find that there's a rough surface. So it depends on the spatial scale that you're operating at and the way you might want to model this. Nevertheless, it's important to recognize this aspect of curvature when we talk about curvatures. So I'll send these papers out in email and in Slack. I know Susan was interested in at least one of them, so.
I think I have it,
Okay.
the one about the sphere.
Yeah.
But yeah, I need to use it as a reference. It's the only thing I can find about why cells are hexagonal on a surface. So maybe it'll satisfy people that I'm working with.
Good luck.
Yeah.
Why are you using a hexagon? And my answer is because everybody else does. Not good enough. It's a low energy situation on a sphere.
Yeah.
There you go.
Why do bees make hexagons?
Yeah. You may have even sent it to me. I don't know, because I get papers from you and Dick, and I try to put them in here, and so I mean, maybe you did send it to me. I don't know.
It ain't
I'm
necessary.
always glad for you to discuss them, though.
Let me show you a fossil.
Okay.
See?
Yeah. Oh wow.
It ain't necessarily hexagonal.
Yeah. What is that a fossil of?
I don't know, some sponge or something 500 million years ago.
Oh wow. Okay. All right.
Preserved in limestone,
Yeah, yes, local.
Oh, okay.
local limestone, yeah.
From the Tyndall Quarry.
Oh, okay, yeah. I know the owners of that.
Oh, good.
Illices, yeah.
Yeah, it's a very nice piece they gave me.
All right. I think that's it for today. Thanks for attending. We had Sushmanth had to leave, and thank you for attending, Sushmanth. All right.
Okay, I'll try to dig out that Turing model paper.
Okay, that would be great. Thanks.
Okay.
All right.
All right, and I'll send you images in a little baggie.
Okay. Yeah. All right. Thanks.
And try for better ones. Anyway, yeah.
Yeah. Talk to you next week. Thank you.
All right, bye.
Bye.
Oh yeah, bye.
Bye.
Thanks.
So Sushmanth inquired about some of the resources we have for lineage trees. And I'll be posting this in the Slack independently of this, but I wanted to go over these in the meeting just to give us some baseline for how we deal with C. elegans lineage trees and determining C. elegans lineages. So as I said in the meeting, C. elegans lineage trees are what they call deterministic. That is that I can take any C. elegans embryo and I can trace out a lineage tree to specific cells, descending from specific mother cells to specific daughter cells. And those cells will generally always have the same function. Now, there are exceptions with respect to defined mutants, but otherwise the scheme always is replicated in every C. elegans. So we can use this when analyzing microscopy images. We can identify cells. They're usually in a certain spatial location. And they also often have later in development attributes of early function. So, for example, you'll have cells that will become connectome cells, or you'll have cells that will become germ cells. And they'll be located in a certain place in a certain sublineage, and then they will eventually have a specific function. So the first resource I want to show is WormWeb. So this is something that was created by Nikhil Bhatla, and this is a resource that's been around for quite a while, at least 10 years, probably about 10 years maybe. So this is an example of a C. elegans lineage tree. P0 is the fertilized gamete, and then AB and P1 are the two cells that divide from that. So the mother cell is P0, AB is this anterior cell, P1 is the posterior cell. And then from AB you have ABA and ABP, so that's a single division. And ABA and ABP are differentiated by their location. So ABA is anterior to the position of AB, ABP is posterior to the position of AB. But both of those cells are on the anterior pole of the embryo. So the AB is the anterior lineage. Now these cells migrate around a little bit, so they don't actually end up in the places that they sort of originate in the embryo. So sometimes you'll have AB cells that contribute to muscle that end up going to the posterior end of the organism, but started in this anterior pole of the embryo. So that's important to remember. Nevertheless, you can identify these cells and track them as they move around. ABA further divides into ABAL and ABAR. So this is now a division of ABA. So this is at the anterior end. It's the anterior division of the anterior end. And then that divides into a left cell and a right cell. So ABAL is anterior left, ABAR is anterior right. It's just subdividing these regions, or it shows that that cell is subdividing either anteriorly, posteriorly, or left to right. As we saw with the respect to the planes of division, we have two planes of division here, left and right, anterior, posterior. Then we have for ABP, which is AB posterior, we have ABPL, which is posterior left, and then ABPR, which is posterior right. So at that second division, they're dividing left to right. So you can see this forms a nested tree. You have nesting. I can open up ABAL and I can see how this proceeds. You have another anterior division after that. You have another anterior posterior division after that. You have a third anterior posterior division, and so on. So they just keep expanding in that way. And their initial position is to be anterior to posterior. But as I said, these things can migrate after they divide. The point, though, here is that they divide, so you can trace it from a single cell down a number of divisions, and you can roughly locate it. It doesn't give you the exact positional coordinates, but its origin is in that part of the embryo. Now, as you'll see here, there's a scale for time line here. The fertilized gamete is at 0. Actually, I think it's like the 2-cell stage that's at 0. And so then we go down to 100, which is where we get this AB, AL, AAA, basically the 5 divisions from AB at about 100 minutes. And then this just keeps proceeding downward, 100 minutes, 200 minutes, 300 minutes. All of development within the egg happens in about 600 minutes. So it's actually a pretty quick process relative to, say, like mammalian embryos or some other type of embryo that we might be more used to. So this is where we get, so these are what they call developmental cells. And they're not stem cells, they have a deterministic fate, but they're still developmental cells in that they're not differentiated into their final form. So they're able to divide and produce daughter cells. And then at some point you get these differentiated cells. So AINL is a type of cell here. You can see at the cell fate map here, it's a neuron or glia-like cell, AINL. It's part of the connectome, and it differentiates at about 350 minutes. So we eventually go from these cells that are undifferentiated to differentiated in terms of function. But these cells were always going to become this cell. So this ABALA, or whatever it is, will become AINL. It just needs to get to that stage. And so at that division, it becomes differentiated. What's interesting in this case is that you get this neuron forming, but the daughter cell to it is what we call sort of an apoptotic cell or a dead cell. And so you get this, what we might consider to be an asymmetric division, where you get one division of one daughter cell becomes a neuron, the other daughter cell dies. And this side we have ABALP that divides into another neuron, and then the daughter of that dies. So you see how that works. You get these divisions of developmental cells, and then you get this differentiated cell where the cell stops dividing and becomes functional. And then there's some differentiation once it becomes a neuron in terms of like the things that are going on inside, like the different types of proteins it's producing and so forth. But it's largely this type of cell. It's not going to change its fate. Now this is all AB cells. AB cells form different things like different types of tissues, like cuticle and muscle and neuronal cells and other types of cells. There is the other lineage. So AB is the lineage that produces a lot of the somatic cells, a lot of the different organs. P1 is more specialized. P1 is a lineage that contributes directly to the germline. So you see that the germline is, of course, where the gametes are produced. And C. elegans is most of the time a hermaphrodite. So hermaphrodites are, can self-reproduce. They have both male and female sexual reproductive organs. But those are all part of what we call the germline, which are these cells that are sequestered away early in development to protect them from mutations. So we have P1, P2, and then this P lineage, P4 goes out to the Z. And the Z lineage or these Z divisions are germ cells. So these are germ cells. They're not shown to divide here, but they keep producing germ cells, and so they produce the reproductive cells. Within this P1 lineage, though, you have other specialized lineages. You have EMS, which I think it contributes to the gut, and MS and C and D. These are all very specialized for different parts of the organism, different tissues. And so this is the second lineage. This is the posterior lineage. So this is all these divisions happen posteriorly. And for MS, for C, and for D, which kind of derive from P1 ultimately, they have the same nomenclature situation as AB. So you have AL, which is from E. You have an anterior division and a leftward division, and then an anterior and rightward division. So that's how that works. And it's the same nomenclature as AB. And eventually, these cells will also lead down to differentiated cells. So E divides until you get to intestinal cells. So these are intestinal cells that now, in this case, you have intestinal 2D, which is terminal. That means it differentiates into this cell, and then it stops dividing. In this case, intestinal 5R actually divides into 5RA and 5RP. So you have this division even when you identify it as a terminally differentiated cell. So it's not always the case that terminally differentiated cells don't divide, but in general they don't. Sometimes you get a precursor of some type, and then you get the final cell. And so this is, again, you see this again with intestinal cells and some of these other specialized cells in this region. So that's our lineage tree. That's a map. It's an interactive map. It's at wormweb.org/cell lineage. So you can look up any cell and get information about it, information about when it divides, what its lineage name is, and then there's a Google Scholar link for things that cite this. So, people have worked on the lineage tree. They've identified specific cells. There's a biology literature on this. People are really obsessed with drilling down to very tiny details in the biology literature. So you may want to check that out. So there are a couple other resources I wanted to go over. The first one is this worm atlas. So the great thing about the C. elegans community is that it has a lot of common community resources. The first of these is Worm Atlas. Well, the first one was the worm, this one here, the wormweb.org. But the second is Worm Atlas. And this is a database that gives us information about anatomy of C. elegans, but also other nematodes. This article is on neuronal cell lineages in the nematode, and so in the nematode C. elegans. And so this was by John Sulston. Now, John Sulston was a scientist who won the Nobel Prize for discovering this lineage tree, and he was the one who actually worked it out by hand. So he sat by a microscope back in the 70s. He sat by a microscope, he drew all these images of C. elegans. He worked out the lineage tree, and then he drew out the lineage tree. So all that nomenclature is from John Sulston's work, and he did this all by hand. So this is a very important set of things to get some background on. So this basically talks about that one of the attractive properties of C. elegans is that we can trace this cell lineage tree, and we can understand how cells sort of form and differentiate in development. And C. elegans is a very good model system for this. So it kind of talks about the nervous system in particular. So, we want to know sort of how the nervous system forms. So the nervous system in C. elegans and the adult consists of this centralized area in the anterior end with the ring, the nerve ring, with the pharynx and some other components. And then there's this tail component, which are a set of muscles and ganglia that control movement. So there are other things in between. There are motor neurons and the ventral nerve cord, which goes up and down the organism, and that also contributes to sending signals to the muscle for the worm to move. Now, all these cells have to divide from somewhere, and so they divide from these developmental cells. And we have this is a breakdown of the cell lineage. So like I said, AB features mostly the hypodermal cells, the neurons, the muscle. MS has a lot of things. MS does have some neurons, some choliomyocytes, muscles and glands. The sublineage E, which we saw, is intestinal. C has hypodermis and neurons, muscle. D has muscle, and then P is the germline. So you can see that muscle pops up in a number of sublineages, and it's just because it's all over the worm. It's a predominant thing, and it connects to the connectome, nervous system connectome. And so, we're interested in looking at how that nervous system originates, how it hooks up to the muscle, how all those things form. So this is the Sulston tree. It kind of shows how he mapped this out. You get these repeated sublineages. You get these post-embryonic lineages, which we haven't talked about yet, but are cells that will divide after the worm is hatched and is actually crawling around in its larval state. And so you'll get different types of cells that are born then. And actually, those kind of cells, you can trace out the cell lineage, but it's not quite the same as what you see in the embryo. So that wormweb resource actually does show some of the post-embryonic divisions and how they end up. So that information is in the wormweb atlas as well. And so they talk about migration, formation of the sensilla, and they show the fact that some of these cells migrate around the embryo, and then at hatch you get a fully functional worm, but you do get some post-embryonic development. They talk about cell-cell interactions. They talk about these different cell lineages, which can be analogous. So this is worth going over if you're at all interested in how, this was sort of developed, this lineage tree, and how these cells function. So we have the atlas from WormWeb. We have this article from Worm Atlas. this is information about John Sulston. This is the Nobel lecture that he gave. So in 2002, he won the Nobel Prize. He talks about, the model organism C. elegans. So C. elegans is a model organism originated with the work of Sidney Brenner. That was in the 1960s. And so they were looking for a nice model organism for medical research as some sort of workhorse. They've used things like bacteria, but they wanted a model organism that had, organs that was somewhat similar to what you would find in humans, with muscle and brain and, other tissues that we can look at. We can look at how those things develop. We don't want to have a lot of complexity, but at the same time, we want to have those organs that are distinct. And so C. elegans became the main candidate for that. So Sidney Brenner worked out some of the first. You have to culture these. You can find C. elegans in the soil just about anywhere on Earth, but you have to culture them in a laboratory, and that's a whole different set of challenges. So Sidney Brenner worked out how to culture these things in the lab, did some early experiments. He was another thing you can do with them is you can induce mutations and have what they call defined mutants, and it's rather easy to make defined mutants in C. elegans as opposed to in other species. But defined mutants are basically mutations that are stable across generations. So you can always later on, when we had genomic data, we could localize these mutations to specific points in the genome, to specific genes, and then have a library of these. So we can actually do experiments on specific mutations and see what the effect is. So Sidney was working on some things having to do with neurotransmitters, and so did John Sulston. So they did a lot of things with, they did some early genomics, which was mainly like sequencing genes and getting, information about some of the structure of the genome. So we have at this point, we have a very good handle on C. elegans genomics. So you can easily map out some of these things, some of these mutations to the genome. You can also understand, which genes are controlling which behaviors. In C. elegans, that's actually easy to do. In other organisms, it's less easy to do, especially with respect to movement behaviors. We know that specific cells will contribute to specific movement behaviors. If we knock the cells out, we render them inoperable. We can change that circuit. We can also do that with deleting certain genes. So this is a very nice model system for probing biological processes. Now he talks about then how he got into development and some of the reasons for that, and how he worked out. So he was drawing these neuroblast divisions, and this is the level of what he was doing. So he was drawing things out by hand, and so this is all very hard work. And if you're interested in this history, but also some context, this is a good thing to read. This is from nobelprize.org. Then there's this article from, I think it's the journal Genetics, and they talk about Horvitz and Sulston on C. elegans cell lineage mutants. So this is Horvitz and Sulston, and they're talking about, Horvitz is another scientist, Robert Horvitz, who was working on C. elegans as well, and they talk about this idea of cell lineage mutants. So you have these cell lineages, and one of the things you can do in cell lineages is you can knock out cells during development. So say if you knocked out this ABALA cell, you would not have a replacement for it. So everything below it in the cell lineage tree would not exist. So if you knocked out ABALA in development, you just ablated the cell, blew it up, you would not get a bunch of neurons. And the consequence of that would be, of course, that you wouldn't have those neurons, and you would probably have a non-functioning nervous system. Other cells you could knock out, and they have different effects. So it's a lot like mutations. You mutate something, sometimes you get behavioral deficits, sometimes they're missing cells in development. You can also do this with getting rid of the cells in development. And so the point is that you can really see what genes are necessary, what cells are necessary, and you can really do some interesting studies. So this is what they were doing with So lineage mutants. It also shows, the level of replacement in the cell lineage. So in a lot of the mammalian cell lineages, the stem cells proliferate and then are interchangeable. So you can put together a cell lineage tree from stem cells that differentiate into different types of cells, given they get these chemical signals and they differentiate. And C. elegans, it's not like that. It's controlled by the genetics, and you can't replace, once you knock out a cell, you can't replace it in the developmental order. So this is a unique system to look at the effects of that. And so they talk about this a little bit here. It's not very long. The combination of molecular and genetic analysis of the cell lineage contributed to our current understanding of the mechanisms controlling induction and development of the vulva, the core machinery of programmed cell death, which is apoptosis, which we talk about all the time in this group, the development of the muscles and nerves that control egg laying, and developmental timing, which is a part of genetics, and the consequent discovery of what we call microRNAs. So C. elegans has been useful from a number of perspectives in studying development. And it's important, I think, that you get an appreciation for this. So this is from the journal Genetics, and I'll put this in the Slack channel for everyone. This is an article by Solston. This is the neuronal cell lineages one. And then we have this paper on cell identification and cell lineage analysis. This is by Andrew Chisholm and Claudiu Geremescu. This is, it's a longer article on sort of the idea of identifying cells in a cell lineage. So this goes through sort of, the rationale. You're looking at cell division patterns, you're looking at gene expression patterns, and how do you identify these cells? Now, in our group, we don't do gene expression analysis to identify cells. We're interested in identifying them using microscopy, and you can do this. It's a little bit different than what they're talking about here, but I think this gives you some background into how these cells, we actually know what these cells are and how we know that they're not, how we know that it's a deterministic process. So we basically confirmed Solston's lineage tree with these type of data, with this type of cell gene expression data. And basically what happens is you can look at some genes that are what they call upregulated or produce more copies of RNA at certain times in development, and that gives them this sort of ability to differentiate into these different types of cells, whether it be intestinal cell or neuronal cell or germ cell, although germ cells don't differentiate. So, we can use something called a green fluorescent protein, and if you've seen these fluorescence images, it's like a black background with like some green or white color that shines, and this is what or sometimes it's red or even blue. This is what we call fluorescent proteins, and they use them to express them in cells, and they may tag like a protein of interest or an mRNA of interest, and they'll co-express so that you can identify these things in an image. So we can get images that have these different GFP markers, and they can tell us about which cells are which. We can also do this through positional information, but that's a little bit less precise. And so these cell identification markers give us a little bit more precision in identifying these different cells. And especially if you're looking at questions of what is a cell expressing, and is it consistent over time, and maybe what are precursor cells expressing versus differentiated cells. Now, for mere identification, we don't necessarily care about that, but this is something that can be useful for other studies. And so if we're creating a tool, say, to analyze cells, that might be something we might build into it. If you're answering specific biological questions, this is also very important to have in terms of a capability. So we can look at comparative studies, which we've done in the group. You can also look at within a specific lineage tree to see what's expressed where. So this is a good article for that. It's very lengthy, but it gives you a lot of background, I think, into this area. finally, this article on a lineage resolved molecular atlas of C. elegans embryogenesis at single cell resolution. So this is a molecular atlas, and this is actually related to some of the work that's been done on the EPIC data set. So this is actually one of the same group, or some of the same people involved in producing the EPIC data set. Robert Waterston and John Murray were involved in that data set, and perhaps others I don't remember, but this is the same group. And with the EPIC data set, they produced information about the different, they were able to produce these movies of the entire C. elegans embryo that's dividing, and the divisions go up to the hatch of the egg. And one of the things I mentioned is that we want to look at molecular signatures to identify these cells and identify when they're going to differentiate into differentiated cells. And so what they're doing here is they've built a lineage resolved molecular atlas. So they've built an atlas that is resolved, the cells are resolved in terms of their position in the lineage tree, and this has single cell resolution. So this has 86,124 or 86,024 single cells, the transcriptomes of these cells. So C. elegans only doesn't have that many cells by far. It has 959 cells in the hermaphrodite, but we use like a number of cells from different C. elegans. We want to take a number of specimens and average it so that we have some idea of the variation. We don't want to just assume that things are deterministic when maybe they're not. Maybe it's just that one worm that you're studying. So they've taken a large number of transcriptomes from a large number of cells, and then they're classifying these cells developmentally into these different categories. And we only have 959 in the organism. And the great thing about C. elegans is it's always the same number of cells per, unless you have a defined mutant, which you can have maybe a few fewer cells. And males, of course, there are 1,024 cells. But the development within the egg, development always has the same number of cells, unless again you're dealing with a defined mutant. The becoming a male happens in the post-embryonic phase. So this is actually a classification problem. You look at these expression profiles, you get a classification of what cell it is in development, and then hence you can figure out where it is in the lineage tree, and you can see which genes change their regulation throughout the lineage tree. That's the idea here. And so they can do this through single-cell RNA-seq, which is a next-gen sequencing technique. And they've done this with C. elegans embryos. They've been able to process this data set and get some answers here. So they have some, you can look through this paper to get a sense of what this landscape looks like. You can get a sense, look at the atlas and see if it's useful. It will definitely tell you more about the lineage tree. In terms of what we're doing with image processing, it may be less useful, but it may also be, something we can use as sort of auxiliary aid in making some of these decisions. So I don't want to bound you in terms of your ambition, but I will warn you that this kind of data is very hard to work with unless you really know what you're doing. So I wouldn't spend a lot of time trying to figure out the perfect way to incorporate transcription data. I would just assume that, if we can make a quick inference from their data, from their atlas, that's great, but I wouldn't get too deeply into this. In any case, I thought I would show you this because it's an important piece of the puzzle. So that's all we have on lineage trees. I hope you found this useful and learned something.